DNA denaturation, as the name implies, something leads to a new state in DNA, which in DNA would not be in its own natural state. Some conditions can disturb hydrogenic bonds. As we learned, hydrogen bonds are in the central part of DNA connecting two base pairs. These two base pairs are from each DNA strand. As you see in this graph, when these hydrogen bonds are disturbed, two strands get separated, and it will be two single-stranded DNA. You know that hydrogen bond is a weak bond, particularly comparing covalent bond in the backbone of DNA. I mean phosphodiester bond, which is not affected in denaturation. The question arises here is that under what conditions DNA gets denatured? When pH gets more alkalinized, when the concentration of some chemicals like formamide or urea goes up, and finally and possibly the most important one, when the temperature increases. Let's say that we have a double-stranded DNA and we change the condition by more alkaline pH, chemical agent, or heating, which converts the double-stranded DNA to two separated single-stranded DNA. And certainly we can reverse the system. If the denaturation condition is removed slowly, DNA can be renatured or annealed. We have two complementary single-stranded DNA. If they were heated, we can cool them slowly. In this way, complementary strands can be base pair again, and double-stranded DNA is produced. This renaturation is a technique which is used in certain blot and PCR. We will talk about the detail of this technique later on. Don't worry. As denaturation and renaturation are important, let's have a review on what happens. As we see here, we have a double-stranded DNA. As the temperature gradually goes up, hydrogen bonds weak, and since AT rich areas are not so strong as CG areas, first areas rich in AT are separated. At higher temperature, CG rich regions also separate. When gradually it gets cold, complementary regions come together and base pairing starts. And finally, renaturation is complete. When we talk about certain blot and PCR, you will see how important it is in order to understand that concept. I mean the concept behind PCR and certain block. Let's talk about DNA organization. All we know that DNA should be organized in order to be packed. But why? The fact is that the huge molecule of DNA should stay functional as well as being placed inside nucleus. So, It requires packing. Since prokaryote cells do not have nucleus, like bacteria, E. coli, it should stay inside the cell. So the amount of packing that it needs it's a bit different, and the way of packing is also a bit different. I mean, in eukaryotes and in prokaryotes. Supercoiling is a well-known method for organizing DNA. As we will describe, you can see why this method is most commonly seen in mitochondrial DNA of eukaryotics, also in most prokaryotic cells. Generally, this is commonly seen in closed circular DNA. Let's remind you that this method also occurs in linear DNA like what eukaryotic cells have. But this is not a prominent method in eukaryotic cells. On the other hand, inside the cells of our own body, we have nucleosome and chromatin in the nucleus of our cells in order to pack the DNA in our chromosome. The fact is that usually nuclear DNA are packed in this way, and our DNA, like other linear DNA, is packed in this method. Let's see the way circular DNA gets organized. This circular DNA is either supercoiled or relaxed. Relaxed circle is like this. Remember that all this pink circle is double-stranded DNA. When it turns around itself, it makes supercoil. Let's have a look at the structure of supercoil to the extent that we need for step 1. Double-stranded DNA twists around itself. It will be under or over winding, the double helix, and it produces supercoil. Please have a look at this picture. When the world didn't have this amount of wireless system, most phones had this kind of supercoil. I think this is a really valuable picture to show the concept of supercoil. I hope with these pictures we can understand the concept of supercoil very easily. As you see, double helix has to turn around itself, and when it turns again, it makes kink or coil of coil, which are called supercoil. We have positive supercoil with which DNA is wound tighter than bDNA, negative supercoil which DNA is wound looser than eDNA, and the very important point here is that negative supercoil is required for most biologic reaction. Let's see how they look like. Remember that negative supercoil is loose and for biologic reaction is used. And positive supercoil is very tight. We saw before relax and also positive supercoil. Here we want to see negative supercoil. And by negative supercoil, I mean this part. As we said, this is looser than bDNA, so base pairing are more accessible for enzyme and biologic reaction can go on easily. So when double-stranded DNA has kink, what would be the way of making the bases of DNA exposed to enzyme? answer is another enzyme or family of enzymes called topoisomerase. This family of enzymes make base sequence in DNA more accessible for other enzymes. The way they do it is by temporary cut in DNA strand by cutting and resealing backbone. In this family, we have two main groups. Topoisomerase 1 simply cut one strand of double-stranded DNA, relax the strand, and reseal the strand. Topoisomerase 2, on the other hand, cut both strands of the DNA helix simultaneously in order to manage DNA tangles and supercoils. Let's see what happens. This is double helix and you have topoisomerase 1, the way it acts. The arrow shows the place it will be cut, and the lower arrow shows that red strand is over blue strand. The cut has been done, red strand still over blue, and finally it should be on the contrary. Turning around the other strand, the red strand is going the back of blue strand. Red strand is on the back of blue, resealing happens, and the red strand is on the back of blue. New arrangement is like this, and the bases here are more exposed to other enzymes. We talk a lot about topoisomerase 1. I think considering what has been said, we can easily understand what's going on. First, the red strand is over the blue strand, then the red strand gets cut, and after that it is resealed, but on the back of the blue strand. And this is the final result of the effect of topoisomerase 1. I would like to remind you again that what we talk here is in the scope of step 1. It could be a bit more complicated than that, but we don't need more complication in the way that these enzymes are acting. These supercoils should be removed. Topoisomerase 2 will do that. But how? Consider this part of the kink. It has a structure like this. In fact, each line here is a double helix. Let's have a closer look at the exact place this double helix cross over another section of its gigantic molecule. It would be something like this. As you see, double helix of red-blue on the top is over the other double helix, and in the bottom is under the other double helix. This is the main reason of the supercoil. Let's see what topoisomerase 2 does in order to solve the situation and make it easier. Topoisomerase 2 cut both strands of the DNA helix simultaneously in order to manage DNA tangles and supercoils. As you see, both strands are cut. Let's watch the rest. And finally, They already sealed the game. So, we realize the way that these sequences of DNA containing supercoil under the action of topoisomerase 2 are changed to this one. The number of supercoil is changed by this enzyme. Also, you can see why in E. coli DNA gyrase or DNA topoisomerase 2 can introduce negative supercoil to DNA. Remember that in E. coli topoisomerase 2 is also called DNA gyrase. I hope here you have a better understanding of the way that topoisomerase 1 and topoisomerase 2 acts. Let's have a look at nucleosome and chromatin to the extent that the exam requires. We will talk, then we will explain them through some slides. In eukaryotes, chromatin is double helical DNA associated with histones and non-histone protein. The basic packing unit of chromatin is nucleosome. Histones. Histones are rich in lysine and arginine. Lysine and arginine have positive charge. The fact is that there is no histidine in histone. Two copies of each histone, H2A, H2B, H3, and H4, form histone octamer. DNA around octamer plus octamer itself make the structure of nucleosome. A series of nucleosomes called beads on a string, they are also called 30 nanometer chromatin fiber. Histone H2A, H2B, H3, and H4. Imagine something like this is H2A protein. And this soccer ball as histidine 2B protein. This ball as H3 and this ball as H4. Remember, they are all protein with positive charges because of lysine and arginine. Let's see how they make histone octamer. As we said before, two copies of each histone H2A, H2B, H3, and H4 form histone octamer. Let's imagine that these balls are arranged like this. As we know, there are histone H2A, H2B, H3, and H4. And as we said before, important point to remember is that they have positive charge because of lysine and arginine. Let's do like this. So, here we have a better imagination. Positive charge around this octamer because of lysine and arginine. Certainly you remember what we said about nucleosome. DNA around this octamer plus octamer itself makes the structure of nucleosome. So, let's turn DNA around our histone octamer. As a matter of fact, this is double-stranded DNA and it turns 2 times around this octamer. You can remember that the backbone of DNA is phosphodiester bound and it has negative charge because of the phosphate group. Considering positive charges of histone octamer, different electrical charges will secure the establishment of nucleosome. Histone H1. Let's consider this stone like this ball. Histone H1 is associated with the linker DNA found between nucleosomes help packing them in 30 nanometer fiber. A series of nucleosomes called beads on a string, also called 30 nanometer chromatin fiber. So, let's see what should be the imagination of that. H1 comes to attach the nucleosome. The linker DNA is the DNA which is located between nucleosomes. In this slide, we can see histone octamer, double-stranded DNA 2 times turns around it, then histone H1, and then linker DNA. In 2 other slides here, you can see how these items are organized. Let me remind you that DNA is considered 2 nanometer and a nucleosome is considered around 10 nanometer. We can see histone octamer, DNA around it which turns 2 times, histone H1, and also linker DNA. I think in this slide we need no more discussion. Just tell you that core DNA means DNA around the histone octamer, and we can see linker DNA here, histone octamer, and histone 1 or H1. You can see here beads of string which makes 30 nanometer fiber. DNA is wrapped around histone. Core DNA means histone octamer, and we will see how they make 30 nanometer fibers. Here we see the way DNA is packed and size of each item. Here you can see histone octamer, and these yellow items are H1 or histone 1. The size of linker DNA is considered 2 nanometer. In this figure, you can see why we call nucleosome the basic package unit of chromatin. The size of nucleosome is considered 10 nanometer. The size of linker DNA which connects nucleosome is considered 2 nanometer. Finally, the collection of nucleosomes makes 10 nanometer fibers. Another slide to show the progress of condensation. More condensation goes on until finally form the chromosome, which in G0 or G1 contains one linear molecule of double-stranded DNA. You can see double-stranded DNA here. Then histone protein is attached. This figure does not explain octamer and H1, but you can recognize them. You can see the nucleosome here, which are the gray color ball with DNA around it, and they got compacted more and more. Please pay attention that in G1 and G0, most of the genome is silent and in compact form. You should know that we have higher order of packing, but for step 1, we do not need to know them. Cell in interface contains 2 types of chromatin. Euchromatin, more open and available for gene expression, and heterochromatin, highly condensed and associated with areas of chromosomes that are not expressed. We know for expression, the base sequence should be copied. So, those bases should be available for enzymes, which are required for copying them, and these bases should not be in compact part. I do really love this figure. I think this is one of the best explanation of organizing DNA in eukaryotic cells. First, we have double-stranded DNA, 2 nanometer in size. Histone octamer is added to it, and nucleosome is formed, which is 10 nanometer in size. When H1 is added to them, they make beads on the string, or 30 nanometer fibers. They are also called 30 nanometer solenoid. They are placed on scaffolding protein in the next step. Approximately up to this stage could be called euchromatin. As we said, this organization goes on, but usually they are not the concern of step 1. This is important to remember that euchromatin generally correspond to the nucleosome. They are 10 nanometer fiber, nucleosome loosely associated with each other, looped 30 nanometer fiber. Let's have another look at this slide. You see, 2 nanometer DNA plus histone octamer makes 10 nanometer nucleosome, which in turn plus H1 makes 30 nanometer fiber. Then scaffolding protein is added to them and it comes to the picture. Another slide regarding that in order to have the best possible understanding of the whole situation. 2 nanometer DNA comes and turns 2 times around histone octamer, making nucleosome. Then histone 1 or H1 is added to this nucleosome, which is 10 nanometer in size. And linker DNA connect these nucleosomes to each other. After that, nucleosomes collect and make 30 nanometer fiber, then scaffolding protein, and finally, as I said, there are something more which is not our concern. So, we come to euchromatin and heterochromatin. We should know that nucleolus is a nuclear region specialized for ribosome assembly. If we are suspected in chromosomal abnormality, we have some options like karyotype and banding. Each of these techniques should be done at a certain stage of mitosis. Karyotype in metaphase, while banding in prophase or prometaphase. Banding is used for aneuploidy, translocation, deletion, inversion, and duplication. And certainly when we come to the part of genetic, at the end of biochem, we will talk about this much more than here. So, let's see which part is euchromatin and which one is heterochromatin. I mean, these two parts. As you can see, one of them is darker and one of them is lighter. The fact is that we can have a very simple understanding, or let's say a very simple rule, which could be just our imagination, but it works here. And the fact is that we can imagine that where the DNA is packed, nothing for imaging can pass. So it appears lighter. And this is on the contrary of active part, which has much less packing. So the darker part is yes. heterochromatin, and the lighter part is euchromatin. So you can easily see why the lighter part is euchromatin and the darker part is heterochromatin. I want you to see some banding technique. In other discipline, they will be discussed more. Karyotype of human metaphase chromosomes. Each human cell contains 23 pairs of chromosomes, which can be distinguished by size and by unique banding patterns. And chromosome banding pattern, and yes, that's it.